Hi, I'm Damon Morelli. Hopefully you know me at this point. This is the fourth video in my Life and Learning Systems series. Today's topic is the coevolution of language in the brain. We'll be looking at the ideas of Terrence Deacon, and most of the information that I'll cover will be from his book, The Symbolic Species, the Coevolution of Language in the Brain. It's a really interesting approach that he takes towards trying to explain symbolic capacity in humans. A lot of individuals have really tried to use the genetic explanation, and certainly genetics plays a role. Yet at the same time, though, I really like Deacon's ideas on the subject. It makes a lot more sense to me. Hopefully it'll make some sense to you. What I'll do is I'll just provide a general overview of his position, and then I'll kind of delve into it. Although I might be a little shallow on the ideas to save time. Real quickly, the overview. Symbolic capacity in humans, and as an extension, linguistic capacity, mathematical capacity, is not a result of a fortunate genetic mutation. Rather, symbolic capacity has its origins in its genetic origins in many of the characteristics that we share with other social mammals, where the real difference comes is that certain behaviors that occurred within a context in the past resulted in conditions that relaxed the role that genes play in symbolic capacity, particularly within the brain, and that these behaviors and the relaxation of the role of genetics actually allowed for an acceleration in the evolutionary process of the brain and of language. So here we have a kind of and this behavior is some sort of proto-language, some kind of proto-linguistic behavior that then allows the brain to evolve more quickly and then allows language to evolve more quickly. So again, they're co-evolving. It's not a linear process. The brain had some sort of miraculous genetic mutation that then allows humans symbolic capacity. So in short, human behavior catalyzed human cognitive abilities beyond the constraints of genetics. It's a really interesting idea. Hopefully it'll make sense when we get to the end of this. So I'm sure that you will agree, excuse me, I'm a little sick. I'm sure that you will agree that humans are very unique, very special. When we compare the size of the human brain to other primate species, according to a brain mass to body mass ratio, the human species is well above average. Here we've got a distribution of the brain mass to body mass ratio on a logarithmic scale, and we can see that humans fall well above where we would anticipate the size of the human brain to be given the mass of the human body on average. Furthermore, the prefrontal cortex of humans is exceptionally large compared to other primates, again, relative to size, disproportionately large. And on a real quick side note, Robin Dunbar has done some analysis of the size of the prefrontal cortex relative to the size of the brain overall of primate species, and has found that there's a direct correlation between the prefrontal cortex size and the average size of the social groups of that species. Again, the idea here is that maintenance of many social relationships requires a very large prefrontal cortex, but also the prefrontal cortex is associated with executive function, and a lot of this function is actually the repression or suppression of kind of more visceral tendencies, more basic primal motivations and urges and behaviors. So again, every time that somebody was maybe angry, if you lashed out and hit people, that probably wouldn't be very good for social cohesion. And again, social cohesion does play an important role in the success of the human species, and of course in language, as we will see later on as well. Humans, very large social groups, a very social species, very large prefrontal cortex, and yet, humans are seen to be very levels beyond other species in regards to our linguistic abilities. Our symbolic capacities seems to be much more much greater in our species compared to other species, and yet our ability to explain these symbolic linguistic differences, these differences in our prefrontal cortex, the differences in the mass of the brain, cannot just be explained by genetics only, as if the genetic differences are not great enough to really explain for these differences or to accommodate for these differences. So how might we then begin to accommodate for these differences? Terrence Deacon seeks to avoid invoking the kind of miraculous, favorable genetic mutation in explaining for the kind of unique symbolic capacities of humans. He terms this explanation to be the hopeful monsters explanation. He kind of lays out a framework for how human language and the human brain kind of co-evolved in a mutual process in which they were kind of mutually reinforcing each other's development, and really allowed for an acceleration in the evolution of the human brain, an acceleration in linguistic symbolic capacity. If we use only a traditional Darwinian evolutionary explanation, we really can't accommodate for the speed at which it seems that the human brain and symbolic capacity has evolved. To explain for the variety of life, the Darwinian evolution uses random genetic mutation as the method for which genes explore and adapt to the environment. Genetic traits thus then become less or more prevalent within a population dependent upon the fitness of those traits within the current environment. And so natural selection then, if these genetic traits fit the environment, natural selection maintains that genetic lineage. If these traits are not conducive to the current environment, well, then these genetic traits are removed from the gene pool. Yet, this traditional Darwinian evolution is a very gradual affair. One of the reasons why is that if you have a genetic trait that provides an individual a competitive advantage, so then it's a competitive advantage that an individual is more likely to sexually reproduce, pass along his or her genes. But at the same time, though, when the reproduction occurs, is that that trait may not likely perpetuate itself through the recombination of the genes. So my point here is just that the process in which genes kind of become solidified within the genome is a real gradual affair, and yet we have this distinct evolution of the human brain that is special, unique. So how can we maybe begin to accommodate for this? And one explanation would be that the environment plays a role, and behaviors within the environment play a role in determining evolution, the evolutionary process. There's Jean-Baptiste de Lamarck. He put forward the idea that behaviors of an individual can then be internalized into the genome and then passed along to the offspring. This hypothesis has been discredited. It's not a valid hypothesis. The only reason why I put it forward here is just this idea of thinking that behavior can somehow affect genes. This is kind of the route that we are pointing to, although we're not pointing to it in a Lamarckian sense. So what we're going to use, or what Deacon uses, is he uses the framework of Baldwin evolution, or Baldwinian evolution, and as well as Waddington's genetic assimilation as kind of as a vehicle for helping accommodate for the role of behavior being kind of internalized into the genome. I'm not really sure if particular in regards to Waddington's genetic assimilation, if it's really necessary. As we'll see, hopefully, in my 6th presentation, which will be looking at Luke Steele's research into the self-organization, coordination of perceptual categories, and a coordinated lexicon. I don't really think that it's necessary. Certainly, the shared genetic characteristics are necessary, but maybe not in the sense that Deacon was looking to kind of put forward a possible hypothesis to support an innatist position. I'll get into that a little bit later. Okay, so he argues though that Baldwinian evolution and Waddington's genetic assimilation can point to the role of behavior in genetic evolution and help accommodate for the rapid evolution of language in the brain. Okay, so the Baldwin effect, or Baldwinian evolution, is this idea that behavior can shape the evolution of a group. And the example that he gives is the example of lactose tolerance. In mammal species, is that once the infant has been weaned from the mother's milk, is that the child then over periods of time develops lactose intolerance, the inability to digest, or the individual ends up loses the enzymes necessary for processing lactose. And yet at the same time, though, we do find that in the human species, in groups with long traditions of animal domestication, animal husbandry, that there is a lactose tolerance among these populations, which, again, we wouldn't expect that given that we are mammals, right? And so an explanation for this is, again, using this kind of Baldwinian effect, is that perhaps a behavior such as, let's say that I domesticate a goat, and I then use that goat's milk, and I consume that goat's milk. Now, that wouldn't necessarily provide for me a caloric advantage if I'm lactose intolerant, but perhaps the milk curdles, some sort of process of creating a yogurt or a cheese that makes it more digestible, is even if I'm not able to derive all of the calories, some sort of advantage in being able to consume more calories than my competitor would then give me a competitive advantage over my rivals. And so thus then in groups that then have established the culture take on the behaviors of domesticating animals, using the animal's milk to create some sort of kind of cheese or yogurt or something like that that can be consumed will provide a kind of competitive advantage, which then increases the likelihood of those individuals within that group of perpetuating their genetic lineage. And so again, here we can see an example of how a behavior can, again, it's not really changing the genes, it's not having a direct effect upon the genes, but it is, the behavior is affecting which genetic group or which individuals are more likely to perpetuate their lineage. So now we get to Waddington's genetic assimilation, and this is more controversial, and I'm not saying that this is valid, and again, as I mentioned before, I'm not even sure that it's even necessary for helping us really understand symbolic capacity in humans. But I'll try. This is a little difficult for me, but I will try to cover this. So the idea here is that there can be genetic adaptations in response to environmental conditions. And then over time, if these environmental conditions persist, is that these adaptations, or perhaps even mutations, end up becoming codified within the genome. And there is some evidence to this that supports Waddington's genetic assimilation. So let's say, for example, that we have a situation in which given environmental conditions are more likely to cause some kind of genetic mutation. And then that genetic mutation confers an advantage towards maintaining one's life, and thus then being more likely to reproduce. Is that then, as individuals who do not exhibit this mutation are less likely to maintain their life and reproduce, is that they end up getting eliminated. They are eliminated from the gene pool. So then we can see how the environment then actually, and genetic mutation then over time will likely become codified within the genome. The example that Deacon uses is the example of sickle cell anemia and resistance to malaria. And it's not that sickle cell anemia is a specific example of genetic assimilation, but it's the environmental conditions, in this case malaria, ends up individuals who exhibit mutations of a specific particular region of the genome. These individuals end up, if they have this particular mutation, they then are more likely to be able to resist malaria. Well, if you're more likely to resist malaria, you're more likely to survive, you're more likely to continue to reproduce, and so we end up finding that this, quote, mutation, although it's probably not even a mutation if it becomes kind of established within the genome, this mutation is prevalent among individuals living in regions where malaria is historically prevalent, regions of sub-Saharan Africa. And these, as well is that these are also the regions, the individuals who are most likely to where sickle cell anemia is most prevalent. And that's just because if you end up getting, you have two alleles within your chromosome, and if you end up getting two of these genetic mutations, two of them within the same within a chromosome, you're likely to you develop then sickle cell anemia, and you don't get the benefits of the enhanced resistance. Now, so in relating this then to language, is that imagine that you have some sort of situation in which, given the environmental, behavioral situation is that you have somehow a symbolic association, perhaps randomly, is established, and that confers a reproductive advantage. The idea here is that if it confers a reproductive advantage, is that then over time even if it's by chance, that genetic mutation matched with the behavioral environmental constraints actually would end up becoming, they use the word canalized. This is kind of formalized within the genome. Again, I don't think that this is necessary, but it's one explanation that Deacon puts forward for support that this would be one way that innatist symbolic capacity could have become formalized within the human genome. But note that this would have had to have occurred very early on within the human species, because all human groups seem to exhibit this symbolic capacity. I suspect that it's probably before even a real delineation of Homo sapiens. It would probably have to come before that, if it were in fact innate. I don't necessarily think it is, but okay. So the shortcomings that we have to the genetic explanations is that there are no cortical structures within the human brain that are particular to humans. The human brain is exemplary, it is special, it is unique. And this actually relates to something Robert Sapolsky mentioned in one of his lectures, but it actually seems that that it has to do with that our genes are just actually formulating kind of two additional neurogenesis, so neurogenetic cycles within our brain. Is that it's not really that we are creating any structures that are unique to humans. It's just we've extended out the neurogenetic cycle. Furthermore, is that the differences in the genetic code cannot alone account for the differences in the prefrontal cortex between humans and other simian primates. Okay, so primates either share the symbolic capacity and, as an extension, the linguistic capacity of humans, or something beyond genetics is playing a role in the evolution of this capacity. And this is where, again, Deacon puts forth his idea of relaxed selection. And this is great and wonderful and beautiful. Okay. So, I will skip over that quote right there. Okay. So, when traits are influenced by external conditions, we are, again, reminded of this kind of inseparable nature of genetics and the environment. And so this is where I really want us to get beyond the ideas of just genetics, just environment, nature versus nurture, is that they really are inseparable. So nowhere is this intertwining more relevant than in the ideas of mind and language. So how might we begin to explain this? Okay, so in some respects, Baldwinian evolution kind of effectively shields particular genetic traits from natural selection by making the genes redundant. Okay, I'll try to explain this. So these genetic traits become relatively neutral in relation to evolutionary processes, and as time passes, the genetic traits are no longer subject to the rigors of natural selection and have a tendency to degrade. The example that Deacon gives here in regards to this is that mammals produce their own vitamin C, with a few exceptions. The exceptions being anthropoid primates, fruit bats, and guinea pigs. Okay, we are we are an anthropoid primate. Why is it that we do not produce our own vitamin C? Well, one of the reasons is that our behaviors and also our color vision and our diet has actually made the need for our body to be able to reproduce vitamin C irrelevant. It's no longer necessary because our dietary habits actually take care of this, meet this need. And this is really interesting, exciting stuff here. So for example, in fact, humans have the remnants of the, and I cannot pronounce this, of the L-glucono-gamma-lactone oxidase gene, the GULO, G-U-L-O, the GULO gene necessary for vitamin C production. We have the remnants of it. However, the GULO gene is largely irrelevant at this point, and as a result, the gene has degraded over time. So now that this gene is no longer necessary to maintaining the life, is that this gene is no longer really relevant to the rigors of natural selection. This is just an example, but keep this in mind when we start thinking about how symbolic capacity might actually be a result of the degradation, the relaxation of the role of genes within our ability to symbolically communicate. So, the role of genetic degradation and its pertinence to language origins comes from the research into the domestication of the white-backed munia, which is a bird. Okay, now this bird has been domesticated for over 200 years in Japan, and it's been bred not for its song, but actually bred for its color. That's an important element. Okay, so so we have the white-backed munia is the wild bird. The Bengalese finch is the domesticated bird. Okay, the Bengalese finch has been bred for white coloration in Japan for over 250 years. The white-backed munia the wild bird, exhibits highly rigid song structures. And again, as is prevalent for wild birds, these wild birds are subject to the kind of competitive elements of natural selection. That these wild birds need to produce very specific songs so as to attract females or attract males, whichever direction it goes. And being able to produce these very specific songs are very essential to being able to reproduce and pass along your genes. Natural selection is playing a very strict role here. Whereas with the Bengalese finch, the songs play no role in its reproductive process. These are domesticated species. They are protected from the rigors of natural selection, right? And so then these Bengalese finches, they exhibit highly variable bird song that is learned through others and through its environment. And I'll quote here from Deacon. Quote, "By removing the stabilizing effects of natural and sexual selection on song production, the almost exclusive control of song structure by a forebrain nucleus designated RAS, I believe, degraded." Okay, so as genes maintaining this behavior template acquired degrading mutations that were not eliminated by selection. As constraints on song generation degraded with prolonged domestication, other neural systems that were previously too weak to have an influence on song structure could now have an effect. These include systems involved in motor learning, conditionally modifiable behaviors, and auditory learning. Because sensory and motor biases can be significantly affected by experience, song structure could also become increasingly subject to auditory experience and the influence of social stimuli. So we have end of quote. So we have a song structure that was highly constrained by the rigors of natural selection. Once you eliminate the rigors of natural selection, then the genes associated with the song structure, as long as the song structure is not the defining characteristic for sexual reproduction, those genetic traits then begin to degrade. As they degrade, then other genetic traits can increase their influence over processes or over behaviors. So whereas in the past the song structure was very genetically determined, now we have a song structure that is less genetically determined, and he gives examples here showing how kind of more complicated these song structures become and are highly influenced by the social environment. You can probably see where I'm going with this then. Okay, so again we have the outcomes for the Bengalese finch then is that we have increased song complexity, greater involvement of social learning and song development, and more diverse neural control of singing behaviors. Okay, so in the past for the wild whiteback munia is that we have this region is primarily associated along with these midbrain regions with song production. Whereas in the Bengalese finch, you can see that these other more outer regions, especially here in the forebrain, which would be for us be likely associated with the prefrontal cortex, more social behavior. Okay, these regions then can expand their role in the process of song, their roles within the neural system, their roles in song production through domestication. Okay, in effect, what we see here is that song production has been offloaded from a primarily genetically influenced region onto more flexible and experientially influenced regions. At some level, the domestication process allowed for enough genetic relaxation for song production to become a more socially coordinated phenomenon. In the process, the neural systems utilized in the production process became more complex, as did the complexity of the production. Further examination of the features of this transition revealed interesting similarities with linguistic communication in comparison to most vocal communication of primates. And I've referenced where Deacon makes that claim. So, in the end, Deacon cautions against placing too much emphasis on the genetic contributions to the evolution of human symbolic and linguistic capacity. Certainly genetics and natural selection play important roles in the process, but recognition of their roles should not come at the expense of interlinked epigenetic factors. Okay, these factors that are associated with how experience affects the development of the brain and then affects behavior. I know that this was probably a little bit difficult. You might have to watch it a second time. Again, I was really trying to fit this within 20 minutes. This is actually my 5th time recording this video. I was interrupted a couple of times, and I think I got faster every time. But again, this is a very interesting suggestion as to showing how the process of civilization and the growth of civilization actually results in a relaxation of the role of genes in the process of symbolic capacity. This is a hypothesis. Hypothetically, could reduce the role of genes and by relaxing the role that genes play actually facilitates or allows social regions of the brain to become more active in the process and allow us to then more quickly assemble, coordinate our linguistic symbolic capacity. This is amazing stuff, and I'm going to try to build upon this in the next 2 presentations, where we will look at the work of Pierre E. O'Dyer and his work on the self-organization, the coordination of shared phonemes, and Luke Steele's work on the self-organization, coordination of perceptual categories and an associated shared lexicon. And again, hopefully we'll be able to then apply this into education and really get beyond these traditional ideas of intelligence and that our cognitive capacities are somehow rooted within our genes, because the evidence is not pointing in that direction. All right, take care of yourself. I hope to see you soon. Feel free to comment on these. I know that I get long-winded. I apologize for that. I am really trying to not be long-winded, but I'm also trying to provide enough evidence so that I'm not just superficially just skimming along things that I then expect you to take at face value. All right, take care of yourself. I'll see you around. Bye.